Good afternoon, everyone. My name is Iman Shah, and I am a first-year PhD student in genetics and genomics, working in Dr. Madan Bhattacharya's lab. And it's my great pleasure and honor to introduce our next speaker, Raissa Fon. She is currently enrolled as a PhD student in botany and plant biology in the University of Yaoundé in Cameroon. She was awarded the US National Science Foundation grant and joined as a research scholar in the University of Illinois at Urbana-Champaign in 2021. She received a master's degree in plant biology from University of Yaoundé in 2014 and a bachelor's degree in biochemistry and molecular biology in 2010 from University of Boya. Raissa has a keen interest in flora identification, systematic conservation of flora. She was trained in plant ecological and morphological characterization during her internship in National Herbarium of Cameroon. Currently, she works with Cylicomorpha solmsii, which is a tropical dioecious tree and holds great promise for use in pharmaceuticals and bioenergy production. Her research focuses on the unraveling the molecular control of sex chromosome in the species via genomics and bioinformatic approaches. Today, she will be presenting a talk titled De Novo Genome Assembly and Sex Determination in Cylicomorpha solmsii. Please join me in welcoming Raissa Fon.
Thank you, all the organizers and sponsors of this symposium. I'm now Raissa Fon, as he said. I'm a fifth-year PhD student, and I'm privileged to present my research project on the de novo genome assembly and sex determination in Cylicomorpha solmsii. So it's intriguing with many here. I work with sex chromosome in a plant species. Some people actually ask me, Raissa, for real, do plants have sex chromosomes? I'll say yes. It's very reasonable when we hear talk about plants, because one thing that comes to our mind is most plants are hermaphrodites, but we still have 5 to 6% of plants showing evidence of separate sexes, that is male and female individuals on separate plants. And I know when it comes to plant breeders, when we talk about plants, one thing that comes to our mind is, especially with multi-omics technology, how can I use my research to contribute to the ongoing global food security issues with crop species? How can I use my research to contribute to environmental degradation that is causing global climate change? But when we look at the human, when we look at the human Y chromosome, we notice that chromosomes occur in pair. And if we have homologous pair of chromosomes, we have to realize that these chromosomes have to have the same size. When we look at the human Y chromosome, we notice that it is very short. It's shorter than the X chromosome. And we ask ourselves, why is the Y chromosome degenerating? It is true that sex chromosome has caught the attention of scientists just in the last 2 decades. And they have empirically proven that the human Y chromosome looked like that of papaya 300 million years ago. What does this mean? It means that the Y chromosome actually is accumulating deleterious sequences that is causing its degeneration. And since plants are thought to have a recently evolved sex chromosome system, any dioecious species is very crucial in understanding the progressive steps that has led to the degeneration of, that is leading to the degeneration of Y chromosome. And we ask ourselves, where does chromosome come from? So there is this convergent model about sex chromosome evolution, that actually sex chromosomes, they originally originate from ordinary pair of autosome in which one pair of the chromosome acquires 2 sex-determining genes, and that is the Y chromosome. And when the Y chromosome acquires these 2 sex-determining genes through a mutation, it becomes linked to the Y and is absent in X. It causes a sexual antagonism. And we all know that during mitosis,
chromosome pairs always exchange their genetic materials. When this sex chromosome is on the Y and absent in the X, there's no recombination in this region. It means the X and the Y does not recombine in this region. What actually happens is that when there's no recombination, the Y, there's a selective disadvantage to the Y. It instead responds by accumulating repeats. And when it accumulates repeats, there is an abnormal size change. And in many cases, the Y chromosome begins to shrink in size. And in some species, the Y chromosome has even been thought to disappear. And the fear is the way the human Y chromosome is degenerating, it might disappear in the nearest future. Okay, one of the purpose of my research was to support this hypothesis in a dioecious Y relative of Carica papaya, since this species is thought to have sex chromosome. This species is a dioecious species. It has a male individual and a female individual on separate plants. And recently, this species has been thought to be a candidate pharmaceutical reservoir. So one disadvantage is that this plant takes 10 years to flower. And at the early juvenile, we can't make a difference between the male and the female. So identifying genetic markers in this species will save us. It's very crucial to separate the species at early juvenile if we need to breed in maybe the females for future research programs. And a little bit of a background about this species. This species is one of the members of the Caricaceae. And in this family, we have 40 species. 38 are actually dioecious and they originate in the neotropical. And we have 2 species that are in the tropical region. We have, in the neotropical region, we have Cylicomorpha solmsii that is in the west of Africa, and we have Paviflora that is in the east of Africa. But when we look at the evolution work that has been done so far in this species, we notice that at least we have the genome size estimated, and only Carica papaya has a reference genome. There is no information, genetic information about this species. Nothing is known about it. So we had to leverage opportunities in multi-omics technology first to generate a reference genome for this species. And reference genomes today, if you go to NCBI database, many researchers would need to pull this, and since it's a very new species, it has adaptive traits that can be used for future research. Sorry, I'm very sorry about this. And secondly, one advantage for this reference genome is that it's a genetic pool for interrogative research, especially in papaya breeding programs. Secondly, we had to use this reference genome for our downstream analysis. First, to fetch out the X and the Y sequences. Sorry, I'm very sorry. We fetch out the X and the Y sequences, and after fetching them out, we look for sex-linked region and look at what is actually going on in the region where there's linkage between the X and the Y, if there's actually degeneration going on. Very sorry about this. This is something else. Next, we have to estimate the divergence time between the X and the Y. And lastly, we identify candidate sex-determining genes in the Y region. And lastly, we had to assess the nucleotide diversity between the X and the Y pair. To attain this objective, or to generate a reference genome first, we had to do a nuclear DNA extraction. We extracted the nuclei of this organism, and after we extracted the nuclei, we had to build libraries. And in the library stage, what happens here is that since we have DNA, like it's a double helix that is coiled, it's very long. So we had to do ultra long read extraction. Now in the next, I don't know what's happening here. So in the library preparation step, we had to, first of all, is there something? Okay. In the library preparation step, we had to, first of all, share the DNA into pieces. That's 15 kb in length. We added adapters so that it can be recognized by the sequencer. We submitted the sequences, our libraries for sequencing. So we did various, we had to submit the sequences for sequencing. We used PacBio HiFi sequencing, we did Illumina, and we had to assemble, we had to also do like HiSeq. Then in the assembly stage, when we got the data, the raw data, that was just like a puzzle. When they give you a puzzle, it's just chopped into pieces. We had to now put these puzzles in such a way that they make sense. And in the assembly stage, what we did is that we took the chopped reads, we assembled them, so we used different pipelines. But the pipelines that stood out was the HiFi. We had to exploit different algorithms. So HiFi could actually give us a deployed assembly. HiFi gave us a phased assembled genome. And after we had to do the context level assembly, we had to move mismatch pieces, that's polishing. And after polishing, we had to do purging, where we phase the genome into the maternal haplotype and the paternal haplotype. Next phase was to annotate the genome. When we talk about genome annotation, we are actually assigning functions to these nucleotide sequences, because after assembly, we just have contiguous sequences, but what is the role of each sequence in the genome? Assigning exons, are there exons, are these repeats? Are these exons? So we had to do annotation. We did a structural annotation and a functional annotation. In the functional annotation, we had to blast our sequences to known databases like the NCBI, SwissPor, ECNOT, and PFAM database. Later, we had to assess the quality of this genome. How good is our assembly? Next, we had to do a comparative genomics to actually see where this organism is from, if it's really a caricature or not. So here we got a phase deployed assembly with an estimated size of 940 kb. That's opposed to carica papaya genome that is half. We got a phase genome with the maternal haplotype. Those are sequences from the maternal chromosome and the paternal chromosome. And we had to assess the quality of the genome based on BOSCO. BOSCO is the number of complete autologs in the embryonic data set. We had a 95% completion, means the genome assembly was good. And we actually identified 40,000 genes in this species. And most of the genome was expanded by repeats. So we had a synteny analysis where we had to do a comparative analysis between haplotype 1 and haplotype 2. That's the maternal haplotype and the paternal haplotype. There was no significant difference between the maternal and the paternal haplotype. Next was to see if this species is actually an angiosperm. So we had to get other angiosperms, and they identified proteins. We saw that this species has shared over 90,000 genes with other angiosperms. We use these autologs. These are core genes that it shares with others. And we also had species-specific genes that we discovered. And when we got these genes, we had to now track the phylogenetic of the species to actually confirm if it is a caricature or not. And we used various algorithms. One thing that worked was nucleotide substitution algorithm in a pipeline called MegaX. And we saw that indeed this species is a caricature and it is a dicot. And this dicot, the ancestor is Arabidopsis thaliana, and they all diverge from the monocot clade, which is Z maize. In the next phase, we now how do we feature out the X and the Y chromosome? In the identification step, we had to use genetic mapping approach. In the first approach, what we basically did, if males have sex, the X and the Y chromosome, and females have XX, what does this mean? We take a deeply sequenced female read from Illumina. We align it to the reference genome. We take the male, we align it. And we use a chromosome quadrant method in which regions in the female that do not align to the male genome, those were candidate sequences that were Y sequences. And we had to use another approach to confirm that. That was a KEMA approach. Actually, we chopped the genome into KEMA sizes of 21 base pairs. We aligned them again to the reference genome, and regions of the female regions of the genome that the female reads could not map, we had to identify those regions as male regions. We proceeded to do annotations. And here, and in the annotation step again, we blasted our sequences to databases, and mainly since it was a new assembly, we had to do de novo, also de novo annotation. We use this pipeline GetAppel. We had to annotate the exons and the introns, and also we identified the sex-linked regions. And the next step was to use KEMA substitution approach to actually estimate the time of divergence. When did the X stop recombining with the Y? So we use different blast approach and different algorithms to estimate that. And the last step of this project was to do population genomic analysis to actually see if there was genetic variation between the male and the females to confirm our hypothesis. So we had to do something like SNP callings, and we used mega blast, blink, fast structure to see if there was a population structure. And the first result was, if we have two homologous pair of chromosomes, what we want is that if they are homologous, we should have to see a curve that is actually homologous. But if we look at the male genome, we saw a kind of heterozygosity in when we had to now, when we were looking at the genomic properties of the male genome, we saw a little bit of heterozygosity that was twice much more than that of the female genome. This was to confirm that all the chromosome pairs in the females are actually homozygous, while there is something going on in the male genome. And this heterozygosity is due to differences in the Y alleles. And this helped us to determine if this species actually has an XY chromosome or a ZW, because in the XY, the male, they are sex determinants, and in the ZW, females are sex determinants. So this was a confirmatory approach that this organism actually has an XW sex chromosome system. And we use also the map coverage to see how well the male reads for map on the reference genome. And we saw that males had a full coverage compared to that of the females. So the next thing was to actually see, look at if there's actually degradation going on in the Y chromosome. So what we did is that we found 30 pairs of genes in the sex-linked region. And we had to look at those genes in terms of gene density. We know there's always, it's now said that the Y chromosome is genetically empty. If it's genetically empty, it means that the gene density in the Y is actually has to be smaller than that of the X, which we found that it was really half, like very small. We had to go to exons. Exons, we all know exons code for genes, while introns are just chunk sequences. In those linked genes, if there's a linked gene, it means that this gene is homologous to its Y counterpart. But we saw that the exon sequences in the Y were far less than that of the X. Means that Y is actually losing exons. When we look at the introns, though it was small compared to the X, we noticed that the length of the introns in the Y were far more than that of the X. And these were those introns that we found that they were transposable elements. Actually, I actually found how they were actually repeats. You can TTAA just repeat, and this CHOM are the ones actually causing the degeneration of the Y. So we could actually support the hypothesis that there's a degeneration going on in the Y. The next was to confirm if plants actually have a recently evolved sex chromosome system. So we had to look for these linked genes again. We had to look for the divergence time. And we saw that it is estimated to be between 0.5 to 11 million years ago. The human Y chromosome system is about 300 million years ago. That's far back. Compared to plants, Silene has the oldest, that is 11 million years ago. So we found that Cylicomorpha also have a recently evolved Y chromosome system. So the next stage was to identify the sex-determining genes in the Y regions. All males differ from females, All males differ from females because males have two pairs of sex-determining genes, while females do not have. And in many plant species, it has been very difficult to fish out these sex-determining genes. But in Cylicomorpha solmsii, in my research, I was very fortunate to identify the two candidate determining genes in the Y regions that could be used as markers. So I identified the mad box genes, and this gene has been identified in Carica papaya as a candidate sex determining genes. And the second one was ethylene transcriptional factor. These genes has not been found in Carica papaya, it's still under investigation. And the next thing was just to do a synteny analysis with orthologs that are found in Arabidopsis and papaya itself to confirm this. So in our next study, we're just kind of like, if males vary from female, we want to look at the genetic structure, because if males really vary from females, the genetic data should actually show that. So we found out that actually there's a population structure in the nucleotide sequences. We had to use different clustering algorithm like Nebo journey and got the SMP data. We found out that there was actually a genetic structure between the male and the female. We also used PCR. We also found a genetic structure, and we did population structure using Bayesian model, clustering model. So we just exploited different clustering models for this, and we found it. And we look at the nucleotide diversity between the male and the female, and we found out that females indeed had a very low nucleotide diversity compared to males. These compared to the males, this could support the hypothesis that males actually have alleles that are homozygous, while males have a heterozygous allele. I want to thank you very much for listening, and I thank my group because I had to work with two teams of researchers, both in Cameroon, that they helped me to actually morphologically characterize this species, and the next group that I worked with was at Urbana, that they helped me in my bioinformatic analysis. Thank you.
Thank you, Raissa, for such an interesting topic. And the floor is now open for any questions.
Thank you for the talk. I think I have two questions. One of them, in one of the slides you showed, there was a priority difference between males and females. And I'm not sure if I understood that correct. Females had one and males had two.
Is this?
Yeah.
Okay.
So do the males and females look different in terms of morphology, or was it just the way the analysis was done?
So it's not in terms of the morphology. Actually, we had to do sequencing of the male genome and the female genome. And we look at the genomic structure. So this was actually a software where we just like supplied, it was Illumina. We did a deep sequencing, I think 70x, and we supplied it to the software, and it had to like, we had this. So this is actually the genetic structure of the male genome. So we had that segregation, that heterozygosity in the male genome compared to the female. Yeah.
The other question I had was the genes that you showed until last slide, the SPP-like and the MATS RBB. Do you know if these genes are also present in other dioecious plants like date palm, for example?
Yes. So the mad box genes is a genes that has been discovered. Like the mad box gene is a gene that helps in, it's a growth gene. It functions in growth. And it's also it's a candidate gene that has been used also in sex differentiation in many species. Yeah. So, yeah, I don't know if I answered your question.
Thank you so much, Raissa.
Oh, I'm sorry for the flight issue.
Okay. Hello, everyone. My name is Sam Blair.